Hi, this is Miss Litton, and this is my wonderful Period 3 AP biologist. Say hi.
Hi.
And we are continuing to discuss our Chapter 5, Membrane Structure and Function, and we're going to pick up in just a minute about active transport across a membrane and how energy is required, but we're going to do a little bit of reviewing first. So, with your bio buddy, discuss this. Why does the salt bother the snails? Have a discussion. Why does the salt bother the snails? All right, so come back to me. When you put salt around the snails, then the environment around the snails becomes hypertonic. And in relationship to the interior of the snail, which would be hypotonic. And water must flow from the hypo. So water is going to leave the snail's body and go out into that hypertonic solution, thus killing the snail. Okay? Good. Ask your bio buddy. Are you right on that? Yeah. All right. Let's see how you did on your individual little reviewing. I'll ask you a few questions. Four number. Try to be in the medal round, Olympians. And by the way, neither AP class has beat 100% in a row. Be that AP class. Be brave in your attempt. Be brave in your attempt. Come on now. All right, chocolate eclairs. It took you 11 seconds to fail. Good job. All right, now she says supportingly. Yeah. Okay. So water's always going to be your what? Solvent. Anything you dissolve in it will be your solute. Look at your bio buddy's clicker. Chastisement or praise. Come on now, you can do it. I believe in you. I got it. Michael! Michael, Oh my gosh. Okay. So, let's review. So here I have a beaker. Okay, and I have something in that beaker. I don't know what it is. Draw. Oh, there it is. Okay. So I have some sort of cell in here. It says the hypotonic solution contains the most solute. Hypo means low. Okay? So if I have this cell and I have, are you focused up here with me? Because it looks like you're not. And if I have this out here, and some more chunks, osmotically active. Okay. So now you would call this solution out here what? Hyper. And you would call this cell, because it has less, hypo. Water must flow from the hypo. So it's going to go from when there's less solutes to where there are what? More solutes. What's going to happen to that cell? It's going to get, it's going to get shriveled up. So the hypotonic solution, by definition, has the least solutes. Hyper. Think hypertonic, hyperactive, too much what? Sugar. Which is really, I don't think true, but whatever. Okay. Chastise or praise your bio buddy. Look at that clicker. Wow, spice cake, peanut butter cookies. Oh no, snickerdoodles has you beat. 5 seconds to fill. Way to go. Sometimes it's a good idea to like draw it out so you can imagine it. Why y'all hassling Michael? Okay, this looks strikingly like our other diagram I just did. Go dots. There they are. Okay, when a cell is placed in a hypertonic solution, okay, so I made this right here. I put a bunch of stuff in it. It is hyper. And when it says something like that, when a cell is placed in a hypertonic solution, then you know the cell has to be what? Hypo. They've already told you. Okay, so the cell is hypo. Which one's going to have the most solutes? The hyper or the hypo? Hyper. Hyper. Okay, hyper always has more solutes than the hypotonic by definition of the word. Okay, so let's look at our choices. The solute exits the cell to equalize the concentration on both sides of a plasma membrane. Okay, so let's first of all talk about that. That would assume that the solute could cross the membrane, right? And then we have something else to consider. It says the solute exits the cell to equalize. Which way would the solute want to go? It'd want to go into the cell. Thank you. It would want to go into the cell because it's going from where it's more concentrated to where it is what? Less concentrated. So definitely not A. Not A because it's not going to want to go in. It's going to want to go, it's going to want to go in, right? Solute, right? It's going to want to go in. Okay, B. Water exits the cell towards the area of higher solute concentration. What do you think? Yeah. Yes. Water must flow from the hypo. So water is going to come out and go here. Yes, B. Let's look at C. Water enters the cell. Is water going to enter the cell? No. So I don't even care what the rest of it says because that's just super wrong. D. Solute exits. Is the solute going to exit? We've already established no. If anything, the solute would go in. So super no. A and C are correct? No. That's like doubly wrong. Okay, discuss with your bio buddy right now and make sure you both understand it. Okay, go. Have that discussion. I got it.
Yeah, there is water, but the hypersolution has
Good job, Pavlova.
Is that you?
Maple leaf cookies. 3 seconds to failure. Nice. Nice. Be so proud. Super fail. All right, clarifying questions you want to ask. Anything you don't get on that. If I was given a question like that on a test, I would always draw it out. Because sometimes I make stupid mistakes. You know, the bad thing is when I make a bad mistake and it's on the key, then all of you miss it. And I have to go back and fix it, right? So I'm just telling you, people can make silly, silly mistakes. Draw it out. Make it make sense to you in your drawing. Then look at your choices. You're more likely to get them correct. Okay? Now, moving on. Okay? So our next part, section, where are we at? 5.3. Active transport across the membrane, energy is required. Discuss with your bio buddy, when we use a protein, is this an example? Make a claim. Is it an example of active transport or not? And support your claim with evidence. Have that discussion with your bio buddy right now. Oldest bio buddy, why don't you take the lead on this? Oldest bio buddy, take the lead. Make a claim, support it with evidence.
This is not bad.
Follow the arrows. Follow the arrows. What? Riddle me this. What would you call the yellow stuff in this example? Solute. What it's dissolved in is the solvent. Okay? So, where is the solute more concentrated? Outside or inside? Outside. Which direction is it flowing? Inside. So if it's going with the concentration gradient, is that active transport? No. What would we call that?
Passive.
It's passive, but you're using a protein, so it's called facilitated transport. Okay? This is an example of facilitated transport because you're using a protein to do diffusion, going from a higher concentration to a lower concentration. This right here would not require energy because we're going with the gradient. Okay? Now, generally speaking, when you do facilitated transport, you're just using a channel, but you could use a carrier. Okay? Generally speaking, it's a channel when you're doing facilitated transport. Yes?
I thought carrier protein always uses energy.
Well, what did I just get through saying? Generally speaking, you would use a channel.
Yes.
Yeah, so but what you want to look at your concentration gradient. If this arrow went the other way, then it would be what? Active transport. Yes?
Just to clarify, diffusion never requires energy, right?
No, diffusion, you're just going from a higher concentration to a lower concentration. Okay? So now let's take a look at this picture right here. Okay? I'm showing you facilitated transport. I'm not calling it active transport. Okay? Youngest bio buddy, tell them why is that the appropriate name for this right here. Go ahead.
Because the molecules want to go in a block, but it's they just slide to the
Okay, so facilitated transport is a good name for this because you're going what? With the gradient. You're going from a higher concentration to a lower concentration. You're just using a protein as a mechanism to do that. Why, oldest bio buddy? Why would they be having to use a protein, do you think? Give a couple of reasons why they would have to use a protein. Go ahead.
They're too large to be both sides. No, I would do that. Look how big it is. You can't just go through.
Okay, what are our two options? Large or charged. If it's large or charged, it cannot go through the phospholipid bilayer, right? What are the three ways? Youngest bio buddy, tell oldest bio buddy. What are the three ways across the membrane? Go. Okay, so what are the three ways? You could go where? Through the phospholipid bilayer, but you can't be? Good. Second way, you could use a or a carrier protein. And third way? Into an exo, and that's when you use the whole membrane. Okay? When I do this, active or passive? Passive. I'm just going from one side of the membrane to another. I'm diffusing right through the phospholipid bilayer. When I do this, I'm a channel, so it's going to be what? Passive. If I do this, it could be passive or active. Okay? If I do this, it's always going to be active. This is always going to require energy to have a whole membrane move around. Okay, check with bio buddies, see if they're in the shadowlands or the sunshine. Guide them if you need to. Be their golem. Eat your frog. Okay, clarifying questions. Okay, so now let's look at this. Whose turn is it, young or old? I'm going to go with this. Light-shirted bio buddy. Make a claim. Make your claim. I'm already telling you what the claim is. The claim is this is active transport. Now support it with evidence. Go ahead.
It says it right there. Okay, for that withdrawal,
Bless you. Okay, so I'm already telling you it's active transport. What's the evidence to support that? Against the concentration gradient, and ATP is required. Okay, for that conformational shape change of the protein, because it's the protein that's escorting it across by binding to it. So you want it to change its shape to bring it from one side to the other side. Okay, now why would you want to do that? Let me give you an example. Okay, maybe you are concentrating hydrogen ions out here. Go hydrogen. Okay, maybe each of these are hydrogen ions. They're really big. Okay, these are supersized hydrogen ions. Okay, now if I concentrate a bunch of hydrogen ions on the outside, this is called forming a chemiosmotic gradient. Super powerful, because there are 3 differences you create when you do that. 3 differences you create by putting a whole bunch of hydrogen ions out here. Can you try and figure out what are the 3 differences I create by concentrating hydrogen ions to the outside? Okay, change in pH, right? Okay, by having a bunch of hydrogen ions out here, this will be a lower pH than the other side. I agree. What else? It's also a difference in what?
charge.
Difference in charge. Okay, this will be a positive charge relative to the other side. And then one more difference. It's the simplest one of all. You guessed the hard ones. Concentration. Thank you. Okay, so a difference in concentration. When you have those 3 differences, charge and concentration and pH, this sets it up like a battery, a difference in charge, and you can use that later to do work. And we're going to talk about that work when we talk about the way a neuron functions and an action potential along the length of that neuron. But we're going to be talking about something else. We're going to, yes, we will talk about it. That'll be the sodium potassium pump for that one. This part right here where you have a concentration of hydrogen ions, photosynthesis and cellular respiration. Cellular respiration in order to make ATP, and photosynthesis as well. You can make ATP by having that difference in charge, and we'll talk about it then. Okay, ask your bio buddy, Shadowlands or Sunshine? Okay, now here is your sodium potassium pump. This picture is too small and lame for you to really see it from there, but let me tell you what happens. You pump 3 sodiums outside of a neuron for every 2 potassiums inside a neuron. So if I pump 3 sodiums out plus charges, 2 potassium charges in, and I'm concentrating them on either side, which one's going to have more positive charges? The sodium side, the outside, right? And you're going to see that with nerve function, because the outside is going to be more positive relative to the inside. The inside is going to have negative chloride ions, and that's going to set you up for an action potential. We'll be talking about that. And also leaky potassium gates, but that's another story. Okay? And I told you this before. 30% of all the ATP you make is what is for this sodium potassium pump. So this is critical for life functions. Okay, your sodium potassium pump, because you're using 30% of your ATP in order to continue to pump sodium out and potassium inside a neuron, for example. Okay, so let's take a look at your notes. Active transport. Molecules are moved against their concentration gradient. It requires a carrier protein and ATP. Requires a carrier protein and ATP. These proteins are called pumps. Is every protein a pump?
No.
Is every protein a pump? No, some are, okay? These proteins are called pumps. Dark shirted one, you take on the example A. Light shirted one, you take on example B. Go. Go.
Sodium is actively transported to our
Okay. Clarifying questions? Okay, you good? All right, now, we said 3 ways to cross a membrane. What were the 3 ways? Through the phospholipid bilayer using a okay, and the 3rd way is exocytosis. Perfect. Okay, and the cool thing about it is this name actually works. Endocytosis, things come in. Exocytosis, things go out. Okay, so this endocytosis, you could be engulfing nutrients. You could be a white blood cell engulfing bacteria. Okay, there are many options here on our endocytosis. Exocytosis, it doesn't mean it's necessarily garbage you're throwing out, though it could be the what's left over in a secondary lysosome after you've digested something, but it could be a hormone that you're releasing out into the body. Okay, so there's lots of reasons why you would want to remove something from that cell, cellular product. Now, on your endocytosis, there are actually three types of endocytosis, and I'll show you all 3. One is cellular drinking, where it engulfs liquid, and it doesn't have to be like just pure liquid. It can have like sugars dissolved in it. Okay. There's phagocytosis with a pH called cellular eating, which would be similar to what a white blood cell might do. And then there's receptor-mediated endocytosis, which is my favorite. Let's think about that one. Receptor-mediated. Remember receptors, one of the protein functions? So on that membrane, you have several receptors that are there, and it's kind of like they land, ding, 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 and all the receptors, got it, got it, got it. And then once they all have it, then it goes, okay, time, and it brings it in. So it's practical, okay, when you do it. So let's look at examples of all 3. The first one is phagocytosis. And could Dark Shirt explain what's going on in this picture? I'm going to give you this one and a real picture. Go. Okay, so on our notes, I skipped over exocytosis, a vesicle formed by Golgi apparatus, fuses with the plasma membrane to secrete substances, such as hormones, neurotransmitters, and enzymes, secrete. Phagocytosis is called cellular what? Eating. Cellular eating. Large molecules engulfed, like white blood cells engulfing a pathogen. Good. Okay, that was dark-shirted one. Yes? Light-shirted? I'm going to give you 2 pictures. Go. Now, penocytosis, a way to remember that, you could say Pinot Grigio, okay? But you wouldn't because you do not drink alcohol. You could think of P, don't drink P, but that should help you with cellular drinking, okay? Pinot, so you get those, don't confuse those. Okay, this could be the endothelium of a capillary engulfing fluids from one side and moving it to the other side. Yes?
So, it can have solutes in it, right?
Yes, absolutely.
So like a solution, basically?
It could be a solution.
So does that mean, like, for example,
Louder, I can hear you.
Does that mean that, for example, with these vesicles, it's like they're membrane enclosed because of this?
They are membrane enclosed vesicles. They are. Okay. All right. And back to dark shirted one. Go ahead. I'm going to give you 2 pictures. Ding, 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 ding. Oh, that's cool. So, penocytosis you already have. Cellular drinking. Vesicles formed around fluids used by plant root cells, as an example. Receptor-mediated endocytosis, a form of penocytosis. Specific macromolecules bind to receptors, selective and more efficient than penocytosis. Selective and more efficient. Yes?
So when the solutes bind with the receptor proteins, is there like a certain process you know? Is just
Wait, say that. I missed it after that. They bind. Now what?
The receptor proteins. Is there a certain process that happens for you to know when they bind, or just know that they bind?
Oh, what you that's all you need to know.
Okay.
Yeah, there's a series of reactions that will trigger, but we're not going there.
Okay.
Yeah. Okay. Good, and good. All right, let's do some quizations. Remember, hundies, people. Hundies. Hundies. Step up your game. This is varsity. Did you know it was back to school night tonight?
Yeah.
You didn't know. Are you going to tell your parents what classrooms to go to so they don't get lost, bumbling around campus?
No, of course they could. They don't want
Oh my gosh. I know. Let's follow the arrows. Chugga, 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 arrow. Oh, now it's here. Chugga, 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 arrow. Now it's here. That looks a little bit like what to me? Exocytosis. Be ashamed. Be ashamed. Show your clicker right now to your bio buddy.
I misclicked, Josh. I misclicked. What time to feed?
Number 1 losers, ribs and pineapple upside down cake.
Oh, that's me.
Ribs can be a dessert sometime, you never know. Okay, what did this colossal fail teach you? Seriously.
Look at the arrows.
Yeah, when you're analyzing a diagram, which chances are you will be analyzing diagrams on your AP exam, this should have been a no-brainer, which clearly it was. You should be able to follow arrows, okay? Practice being smart, not ignorant, okay? Then you're more likely to make good choices later. She says in love. Josh, you should know this. Oh my gosh. Strawberry shortcake.
Johnny's
Oh my God. Who did that? Who did that?
Andrew.
Strawberry shortcake. Not paying attention. Clicked it in. Now, good job. Gold medal ribbon or gold medal for chocolate eclairs, silver to red velvet cupcakes, and ribs. You got the bronze. You're up on the stand. Okay, now, what does the error of this question teach you? What do we learn from this?
Read the whole question.
Read all your choices, right? Read the whole question. Read all your choices before you select. Okay? You just keep that goal of getting 2 hundies in a row, peeps. You keep that goal. I think so.
Totally. That's totally A. Guys, if we don't get 100,
No cheating. No cheating. Shh. Don't yell it out, think it out. Don't yell it out, think it out. I'm going to ask you one little hint. Do you know what a pickle was before it was a pickle? A cucumber. Think about it. Think about it. In another life, a pickle was a cucumber.
B. Totally B. Kid, I didn't put B. Kidding. Guys, don't go for B.
Don't yell anything, Ori. Ori, we know. I made it clear, right? I thought I was clear. That is a cucumber. That cucumber is getting smaller, so it can become a what? A pickle. If it's going to become a pickle from a cucumber, what must happen? Water must leave it. Water is leaving it because what kind of solution is it sitting in? Oh
my God.
Baklava. Awesome. Because that solution is hypertonic. So close. All right. Now, if you look at your group shared notes for the next part, stop and listen. Modification of cell surfaces. The cytoskeletal and cell junctions are not on the essential knowledge what? What does it say? List for AP bioexam. But they do help you understand cell motility and division found in later chapters. So do you see how some of it, like I even put the font super small? That is just there for support reasons. Don't worry about it. They're not going to ask you a direct question about this. It's just for your overall understanding. So just appreciate, absorb, highlight, maybe put some pictures in there if you want to. Okay, so the first thing we need to think about is what easily binds plant cells together? Cell walls. Do we have cell walls? No. So I don't want to be able to rip my skin off right here, right? That would be super sad. Okay. Could I actually rip my skin off? Yes. Okay. So there's something holding those cells together. Well, the first thing we want to talk about is what holds those cells together. So I'm going to jump to number 2, then I'll come back to 1, okay? So junctions between cells, that one's the bigger font, and I'll come back to the other one. So there are different types of junctions, and I believe I introduced these earlier on, right? So you have adhesion junctions, and adhesion junctions is where the be an adhesion junction. Be adhesive. Look at me. You can make an A, adhesion, right? See the A? And A adhesion, and the cytoskeleton is actually extending from one cell to the next cell in an adhesion junction. And here's a picture of that right here. Okay, so the cytoskeletal filaments are just extending, and you have some anchorage there and some protein plaques, but it helps. It's like sharing a skeleton. I think the example I probably did here is if my radius and ulna extended into her radius and ulna, and that would so join us together. Okay, so that's adhesion. You could start by making your A like that to think cytoskeleton, adhesion junction. Okay, your next one I want you to look at, okay, is called a tight junction. And this is when it's protein to protein. So make a tight junction with your knuckles. Tight junction. See how tight it is? Okay, it's one protein connected to another protein. Okay, this is good. It doesn't allow for leakage. Okay, like you don't want things to leak out of your intestines. Your intestines, remember, I think I probably told you this, like it could be more than 30% of the dry weight of your feces is bacteria. Like if you took a dump on a paper plate and let the water evaporate out of it, okay? That's always fun. And we'll get you a homecoming date. Okay. But if you did that, the bulk of that dry weight of your feces is bacteria. And if that got into your abdominal cavity, that would be really bad. That's why if somebody's appendix bursts, you have tremendous complications. You want to get it before it does that because you're releasing bacteria all throughout that. So you don't want it to be leaky, you want it tight. Okay, what was the first one we talked about? Adhesion, tight. Now go like this. Okay? This is when you line up channel to channel. That's super cool because now something on one side can go right through to another because you've lined up your channels, you've docked them together. And that's called a gap junction. Okay, so you have 3 on there. The adhesion junction, mechanically attached adjacent cells using plaques and sharing of their what? What are we sharing here? Cytoskeletons. Sturdy and flexible. Used to connect what kind of cells? Skin. Tight junctions are adjacent plasma membranes stitched together by shared what? Proteins. Used to connect cells of the intestine and other places. Your bladder too has tight junctions. When you say leaky bladder, we probably think, oops, I went a little bit in my undies, right? But I'm talking about the sac that's holding the urine. You don't want the urine to leak out into your abdominal cavity.
Are all cells like connected by junctions, or are there some?
No, think about like red blood cells. So most cells have what's called anchorage dependency. This is a really good question that you asked. Most cells have anchorage dependency where they actually need to be attached to another cell. Things like red blood cells and white blood cells don't, right? Because they're flowing.
Yeah.
But that is a hallmark of a cancerous cell. It loses anchorage dependency, and so it can metastasize and spread to other areas where it shouldn't. Like you would think, okay, skin cancer, it's isolated, it's in that one spot on your skin.
Yeah.
And if it's like a patch of, I'm sorry, I'm glad I don't know, like if I had a patch of skin cancer here and we get that removed, that would be really good. But if I let it sit there, it might digest through the basal lamina, a barrier that keeps my skin separate from my other tissues. If it gets through the basal lamina, and then it can hijack like your lymphatic systems everywhere in your body, your circulatory systems everywhere. It can hijack. It's like getting on a train and then riding to some other part in your body and then getting off the train and setting up camp there. And that's when it metastasizes. It moves to another part of your body. Does that make sense? And it's lost anchorage dependency when it does that. Now, leukemias, you're not even thinking about that because those are white blood cells and they already don't have anchorage dependency, right? And that's why it just goes everywhere in your body. Okay, moving on. We are on notes. Gap junctions. Shared, what? What are we sharing here? Channels. Shared channels between adjacent cells. Allows for communication and strength. An example I gave you there was your heart. Okay, now here we're seeing channels, here we're seeing tight junctions, here we're seeing cytoskeleton. It's not like it's clean and neat between each of these. What it really looks like is maybe something like this. Okay, so there are other items there as well. Here's where your cytoskeleton is connected to a what? Receptor. Okay, so that receptor, it could be used in signaling. So something binds to that receptor and signals the interior of the cell. It could be elastin, and it's all about resilience, elasticity of your skin. Because I am 50, I am losing elasticity in my skin, so that's when you get what? You were supposed to say more beautiful, okay? Wrong answer, fail, okay? No, that's when you start to get wrinkles because you lose that, okay? And then collagen is for what? Strength, okay? So these, you could use it in cell signaling, resilience, strength. There can be proteins out there and other items attached to those receptors as well. And I gave you that, if you look underneath extracellular matrix outside of the cell, that's called the ECM, is the extracellular matrix. Do you want to add that in parentheses? It's a matrix of, and you would have guessed this, proteins and polysaccharides, right? Because you know about carbohydrate side chains, right? Glyco, lipids, and glycoproteins, which are all together part of the glycocalyx, okay, and proteins. And then I gave you some support examples of collagen and elastin. Okay, and I gave you a control example, but I kind of made those small. Look at E. dark-shirted bio buddy, explain 1E about the variability in ECM and how it affects structure. Take that one on. I'm sure you'll have no trouble. Go ahead. So if you put a lot of salts in here that solidify, right, then it becomes like bone, less flexible, right? Whereas collagen is more flexible. That's all because of the ECM. We're going to learn that actually it's not part of the objectives you have to know anymore, but just generally speaking, there are 4 types of tissues. There's epithelial tissues on the outside. There's connective tissue. There's muscle and nervous tissue. Those are your 4 big tissue types. Then when you talk about connective tissue, those are cells that sit in a matrix. They sit in something. Like red blood cells sit in plasma. Bone cells sit in salt. Fat cells, adipose cells, that's connective tissue. They are sitting in a matrix. Sometimes that matrix is very hard, like bone. Sometimes it's very liquid, like blood. Okay, and then that's when your ECM is going to become very significant. All right, next, plant cells. Plant cells have walls, okay? And, hi wall, where are you? Okay, so here is your cell membrane. Here is your primary wall that is laid down. And then, do you see right here where it says middle lamella? That would kind of be like the ECM for plants. You all right with that? And that, do you see where it says pectin? It has pectin in it. And we might have mentioned this before. You know, like say you have some nectarines at your house, but when your mom or dad or grandma bought them, or grandpa or whatever bought them, they were hard, right? And then when they ripen, you grab them and they're softer to the touch, so you know, oh, it's time to eat that. Now, you don't like to do that if it's an apple, right? If it's an apple and you're going smushy and you're like, bad apple. But we do like it for things like nectarines and peaches and avocados. And that's when this pectin starts to loosen up, then we know that whatever's inside of it is a little bit more sugary. And then that's when we like the flavor of it. Okay? Now, here's the cell membrane, here's the primary wall, here's that pectin. Where is the secondary wall going to go? Discuss with your bio buddy. Where's the secondary wall going to go?
I think it's above.
Okay, so I'm going to put 3 places and you decide. Is it going to go here? Is it going to go here? Or is it going to go here? What do you think? Who's going to make it if it's A? I'm just curious. Because that's outside of the outside of a cell. How would it even get to B? It couldn't, right? Where's the cell? Here's your cell membrane, people. Who's going to make another cell, more parts to the cell? It has to be right here. So your secondary wall gets laid down, okay, between the cell membrane and the primary wall. Follow that? Make it make sense to you. Okay, because you're a cell and you have generated that primary wall. You've got to generate a secondary wall to go to the interior of it. Are you getting that? So who has secondary walls? Well, when you have something like grass, it's got a lot of flexibility to it. Now imagine an oak tree. Okay? When they talk about the hardness of woods, it has to do with that secondary wall. Okay? Because wood is xylem. Did you learn that in like 8th grade about xylem and phloem, or 7th grade or something like that? It's just xylem. And depending, like, you know, some wood you can scratch into, okay, and some wood is a harder wood that you can't do that to. So those secondary thickening, all of that's going to happen between the primary wall and the cell membrane. All right, now, if you have, let's say, 2 walls, let's say you're a woody plant that has 2 walls, how's one plant cell going to talk to another plant cell? You have to have a what in the middle of it? A doorway. That doorway is called a plasmodesmata. Okay? And so you know what it reminds me of? It reminds me of a what? Gap junction. Yeah. It's kind of like a gap junction, but it's a hole in the cellulose. It's a hole in the cell walls. So one cell can communicate with another cell. So sugars can be passed from one cell to another. So waters can be passed from one cell to another. Okay, so on your notes, plant cell walls, porous, varies in thickness depending on function. Primary wall, go down to secondary wall, form to the interior of the primary wall. Form to the interior of the primary wall. And then plasmodesmata, narrow membrane-lined channels that pass through cell walls of neighboring cells and connect their cytoplasm. My friends, this is a map. I didn't say it was a man. Multiple answers necessary. I said it was a map, multiple answers possible. But it is a man. About half of a man. Guys, I want you to always have a fire buddy. Somebody you go with, and I recommend it's your bio buddy as your fire buddy, okay? And we have a fire drill, and I would make sure I teamed up, okay? And I want to tell you this right now. If we have a real fire drill, and for some reason Miss Litton gets knocked out, and you don't think I have a broken back, drag me out. Okay? Girls in the front, what are some things you think I want out when there's a fire? You're going to guess. What are some things that I would think are important? I want my car keys. My computer, my phone. Grab my phone for me. It's always, all my stuff's always sitting right here, okay? Take anything that looks heavy or expensive. Phone, yeah. Computer, yeah, my purse if it's here. Critical things, okay? That's in charge. You're like, yeah, got it. Starbucks. I don't think about those. Okay, look at this safety binder, so you know. If something happens to me, okay, let's grab the safety binder because inside of it you will find a roll sheet of which I will be taking roll, okay? You can do that if something happens to Miss Litton. Please tell somebody if something happens to Miss Litton, okay? If your bio buddy, if your fire safety buddy is not moving, okay, don't just go, it's about me, okay? And just leave, okay? We want to, I got you, Josh. Yeah. Yes.
Before we get to the drill.
The drill is not till 11:35.
Oh, okay. Just the 20 minutes. Okay.
Yeah.
Like 10 minutes.
Oh, you're talking about your thing. Run over there, yes. I'm like, no, we're not going to leave 20 minutes early. Wow. 6 of you passed. Hold those clickers high. Nice. That was a hard one. Okay. Let's talk through it. Plant cells always have a secondary wall? No. Who did I say would have secondary walls? Woody plants. Do you see that? Find it in your notes. Highlight it because you missed it. Secondary walls when you have woody plants. Plants with bark. Okay? B. Have channels between cells that allow strands of cytoplasm to pass from cell to cell? Yes. What do we call those channels? Plasmodesmata. Thank you very much. Nice. Okay. Do they develop turgor pressure when water enters the nucleus? No. No, when it enters the cell. Please. Vacuole. Thank you. Okay. Do not have cell-to-cell junctions like animal cells? No, they do. Yeah, we just talked about one. Plasmodesmata. Have lignin to add strength to secondary? Yes.
That was in the notes.
Okay, tell your bio buddy, did you get it right or which ones did you miss?
I missed A, Josh, no, we missed C.
What? Because this says do not have cell-to-cell junctions, and they do have cell-to-cell junctions. Yeah. All right, that is the end. It is time to look at data, and we hope you're having a great day. Be an osmotically active substance.